Hi, I'm Chloe. I'll be discussing the treatment of Alzheimer's through nanotechnology-based drug delivery systems. So first off, some physiology of the normal functioning of the brain. So the brain consists of three main parts. The cerebellum, which is involved in remembering, problem-solving, thinking, feeling, and movement control. The cerebellum, which controls the coordination and balance. And the brainstem, which connects the brain to the spinal cord and controls automatic functions such as breathing, digestion, heart rate, and blood pressure. So the cortex is the brain's outer layer and it has four different lobes. The frontal lobe is associated with reasoning, planning, parts of speech, movement, emotion, and problem solving. The parietal lobe is associated with movement, orientation, recognition, and perception of stimuli. The occipital lobe is associated with visual processing, and the temporal lobe is associated with perception and recognition of auditorial stimuli, memory, and speech. But all these functions cannot form without the work of individual neurons deep inside each element of the brain. There are over 100 billion neurons with branches that connect to over 100 trillion points inside the brain, and it is referred to as the neuron forest. But we'll get back to this a little bit later. Now, Alzheimer's disease is a neurological disorder which results in cognitive and behavioural impairment of the patient. Generally, the cortex shrivels up, damaging areas involving in thinking, planning, and remembering, particularly in the hippocampus, where new memories are formed. This explains why when someone is diagnosed, those newer memories are the ones to be gone first. Now, going back to the neuron forest and looking deeper into this area, the neurons of a healthy brain communicate and connect through synapses, a chemical trigger which allows for one cell to talk to another using neurotransmitters. This is performed in an array of steps. So, firstly, after the depolarized wave arrives at the presynaptic terminal from another cell, the wave causes an opening of calcium channels, allowing for calcium to flow into the cell. This promotes fusion of synaptic vesicles and the presynaptic membrane and secretes the neurotransmitter into the synaptic cleft. This allows for neurotransmitters to then diffuse across the synaptic cleft and binds to ion channel receptors on the postsynaptic membrane. This opens up sodium channels for the sodium to flow into the cell, leading to depolarization and triggering the action potential. The neurotransmitter is then formed in the cell body and transported down the axon to be packaged into vesicles and continue the signal. For those with Alzheimer's disease, plaques of protein called beta amyloid build up in the brain and tangle between the nerve cells, affecting the synapse system, causing them to shrivel up in the cortex. This particular protein is sticky in nature, making them dangerous in groups as they block the synapse signaling and have the ability to trigger immune defenses, inflammation, and swelling. So as tangling continues, the cell transport systems are blocked, meaning that cells can no longer receive the nutrients and water they need to survive, as well as they can't excrete any waste, causing them to die. So these plaques then spread over time, affecting more and more neuron cells. So there are three stages of Alzheimer's. In the earlier stage, symptoms are hard to detect as there are only small areas of learning, memory, thinking, and planning that are lightly affected. An individual could be in this stage for up to 20 years without noticing a problem. And the second stage, if these areas are more heavily affected, and the individual may notice a small change in their brain activity during day-to-day tasks. So here the disease begins to spread and affects an individual's speech, spatial awareness, as well as more severe learning, memory, thinking, and planning abilities. So in the developed region of Alzheimer's disease, most of the cortex has been damaged and the patient loses ability to look after themselves and has severe loss of speech. They also find it hard to recognize family along with other developing problems inside the brain's activity. So these images, the darker purple regions of the brain is where the plaque is more built up. So scientists have had trouble in locating how the disease comes about for prevention. However, studies have shown that advancements in nanotechnology may be able to assist in treatment, reducing the effects of Alzheimer's. So currently, treatments such as acetylcholinesterase inhibitor drugs fail to reduce the effect of Alzheimer's due to their poor solubility, low bioavailability, and ineffective ability to cross the blood-brain barrier. So the blood-brain barrier is a semi-permeable membrane separating the blood from the cerebral fluid and only allows for small molecules, fat-soluble molecules, and some gases to pass freely, while other larger molecules, such as glucose, may enter with the aid of a transporter protein. However, all other larger and unwanted particles cannot pass through. Now, nanoparticles, they hold properties of high biocompatibility and are small enough to hold physical and chemical properties that allow them to cross the blood-brain barrier undetected. So nanotechnology is composed of a range of systems, including polymetric nanoparticles, solid lipid nanoparticles, nanostructured lipid carriers, microemulsion, nanoemulsion, and lipid crystals, all holding individuals' properties which can be advantageous in delivering certain drugs at different speeds to target different regions of the brain and other parts of the body. So, Focusing on liposomes as a drug delivery carrier, studies have shown that this technique can be advantageous in passing on drugs through the blood-brain barrier to reduce the effect of Alzheimer's in patients. Liposomes are vesicles consisting of one or more phospholipid bilayers, and having one layer classified as unimolar, and those consisting of two layers are classified as multimolar. These can be further classified as niosomes, transposomes, allosomes, and phytosomes, each holding different advantages in relationships to biocompatibility, pH sensitivity, and size. In order to increase the possibility of liposomes crossing the blood-brain barrier, they have been functionalized with an anti-transferrin receptor antibody, such as modified cell-penetrating TAT peptide. In order to test these methods, mice were injected with liposomes functionalized with their anti-transferrin monoclonal antibody and dual ligands, otherwise known as MAB and peptide PAA. So the next step to developing this theory is to load the drug rivastigene, which helps to treat mild to moderate Alzheimer's. A model of an Alzheimer's affected brain was prepared, and then rivastigene loaded liposomes inserted. This showed advantageous results as the model could be normalized. This technology is now being further developed to be tested in terms of effective administration and increasing the effectiveness and drug release time for the human brain to then be used in the future to treat mild to moderate Alzheimer's. Thank you for listening, and I hope you enjoyed my presentation.